Hi, everyone. Good afternoon. My name is Zhiyong from the NCB. I am presenting on behalf of one of our trainees from the National Library of Medicine Intramural Research Program. Unfortunately, Dr. Zhiyong, the first author of the work, cannot make it to this meeting, so I'm presenting really on his behalf. So what I'd like to tell you about is how do we improve standard large language models for gene set analysis. So what is gene set analysis? Why is it important? Well, in today's research, there's a constant need for analyzing or interpret data that are coming out of those high throughput experiments, such as microarrays or RNA-seq, where there's a list of genes that are differentially expressed. They are upregulated or they're downregulated. So the computational goal is to determine collectively the function of this group of genes. How do they work together to perform a specific function? So the standard methods that are used by the community is gene set enrichment analysis. This has been extremely successful and has played an important role to drive knowledge, advanced knowledge ahead. But there are also some of the weaknesses or limitations of this type of approach. One is that if we do this kind of statistical analysis, it has to using or rely on the predetermined or predefined gene sets already curated in existing databases. And also, these methods will give you and predict reasonable biological functions, but without telling you how these genes are actually working together to achieve those functions. So in the era of large language models, there has been two very inspirational studies that have already looked into this problem and basically evaluating their capability of large language model for gene set analysis. Now, I came to this meeting, I realized actually these two papers are also being presented at the same meeting at this conference. So I strongly encourage you to attend these presentations as well if you're interested. In particular, the study led by a UCSD group showed that when they compare, basically they give a set of genes as the input to large language models, and they experimented with multiple large language models, and they showed that GPT-4 works the best in terms of evaluation, where they given a set of genes that are related to Go terms, and then they basically ask GPT-4, can you actually generate the ground truth? And when they compared the generated results against the ground truth, they find that 60% of the time, GPT-4 generate output very highly similar to the Go term in the ground truth. And another 40%, there's no match, but 1/3 of the no match is actually matching to something more broader, while the other 2/3 are not there. So given our research expertise and experience, we are really interested is how can we actually further improve these results? If you ask a human scientist to improve this result, what they're going to do is they're probably going to check the literature and check in also the knowledge that are included in those expert curated databases. And that's sort of the idea we took. We have, this is, in my view, is a unique asset of the biomedical domain. Our research field is we have these domain resources that we ought to use them for our domain problems. And that's exactly sort of the technical innovation in our approach called the GeneAgent, where we're adding this additional module on top of the standard GPT-4 output, where we ask it, because it's not efficient or feasible to have human scientists to actually do the checking yourself, because you can't do that for one set of genes, but you probably can't do that for a large set of genes. So what we have designed here is this agent, which is another large language model. It would actually do the self-checking by itself. How do we do that? Well, this is the other box I'm showing here, where it's actually given the set of genes in the output of a GPT-4, it will automatically trigger to using relevant APIs that would actually getting the information, relevant knowledge in the relevant databases. And then based on the knowledge retrieved from relevant databases, it will then determine whether or not the original statement generated by the standard GPT-4 is correct or not correct. If it's not correct, what should be the correct answer. So on the right side is example where we show you here there's a set of 6 genes that original GPT-4 thinks these genes are relevant in RTK signaling. But when we put that into GeneAgent, agent will activate the APIs to go to the relevant database, and then using the relevance, retrieve the knowledge to verify this claim or this statement. And by putting them together, this particular case, the agent or the self-verification module disagree with the standard GPT-4 output and think this is more likely to be MAPK signaling instead of RTK. Now, how did this approach work? Well, we compared on 3 independent benchmarking data sets, because we combined all these 3 data sets that were previously evaluated using in the other 2 studies, which already showed that the method, the standard GPT-4 method, is already as good as the conventional or traditional gene set enrichment analysis. Okay, so what we're trying to here is to show you here in the bottom part, where on these all 3 different data sets, we showed that using the self-verification module, we can consistently further improve the performance by 20 to 40%. So this is very encouraging to us, but we want to know that even though we know GeneAgent, the self-verification module, can help improve the performance, do they actually do the work correctly? Because sometimes even GPT-4 can give the correct answer, but its reasoning may be flawed. So the only way we can check that is we have humans to look at the verification report. So we recruited 2 domain experts. They manually looked at over 130 verification reports, and they determine whether or not these verification is correct or not. And here is the table showing you that, for example, GeneAgent, among the 132 reports, it supported 88 cases. GeneAgent think this GPT-4 result is correct. Well, if humans look at this, 80% of the self-verification report is correct. 6 of them are partially correct, and 2 are incorrect. So overall, there's over 90% of accuracy in this self-verification process. So now, given this very promising results in the offline evaluation, what we like to do is we want to see whether this is something can be applicable to real-world scenarios where we are given novel gene sets. So through our NCBI colleagues, we were connected with 2 domain experts from National Cancer Institute, where they actually are working on a problem where they have novel gene sets from mouse melanoma cell lines. So they give us a total of 7 novel gene sets. So we took those gene sets. We don't know the answer. We put them into GPT-4. We took them also to GeneAgent, and then we give the results back to our domain experts and ask them to judge not only just the accuracy, but in 4 different aspects: the relevance, the readability, the consistency, and the comprehensiveness. So final conclusions are showing here that out of the 7, I think 5 of the 7 that GeneAgent is significantly better than the standard GPT-4. In one case, neither did good. In one case, GPT-4 was more preferred. So this basically along the lines of our research, where we're very interested in evaluating and enhancing standard large language models. But at the same time, we are very interested in the novel applications, and in terms of deploying and implementing large language model in the healthcare domain. Safety is another very big issue in our current research. And recently, we are encouraged because we were selected by the NIH Director's Award for continuing this line of research. And because that, we are able to having additional trainees to join our group. So if you are graduate students wanting to do research at the NCBI, or you are graduate students about to graduate considering doing a postdoc, feel free to contact me either at this meeting or using the email below. So with that, my last slide, I'd like to thank the colleagues who worked on this project or helped with this project. But also, I'll give a good shout out to all these databases, wonderful databases, because if you were at the keynote at the very beginning, I think our study basically is a good evidence of how these well-curated databases can contribute to to advance science, even in the era of large language model. Thank you very much.